G'day guys. Today we're looking at how we can actually reduce calcification in the arteries. Let's get on with it and take a look at what the research says. Anyway, so I grabbed this study, and this is one that was actually done in Germany, looking at vascular smooth muscle cells contribute to atherosclerosis immunity. So just so we understand what we're talking about, vascular smooth cells pretty much constitute the major cells in the media layer of the arteries that are critical to maintain the integrity of the arterial walls. Okay, they participate in arterial wall remodeling, and we want the remodeling to stay in a very, the way it should be, and play an important role in atherosclerosis throughout all stages of the disease. Studies demonstrate that vascular smooth muscle cells can adopt numerous phenotypes. That means different sort of configurations internally, the modeling, depending on inputs from endothelial cells of the intima, resident cells of the adventitia, and circulating immune cells, hormones, and plasma lipoproteins. This plasticity allows them to perform multiple tasks in physiology and disease. In this mini review, we focus on the previously underappreciated activity of VSMC, their impact on atherosclerosis immunity via formation of arterial tertiary lymphoid organs. Now, I'm not going to go into this study because that's not the focus. It's just to give you an idea what we're talking about here. There's basically, these guys are from research establishments in Wuhan and Germany. So, and this is a review. So we're really looking at, if people want to actually go through this and look at it and sort of look at the sort of issues that are associated with the intima and how this sort of, there's a fibrotic cap, and this is the sort of different process that are taking place in calcifying that and stabilizing and putting, and then the different sort of things that are at play, macrophage, cytokines, and you name it. So people can go through this. That's not the focus of this video. It's the focus of looking at strategies, how to decalcify. And so this will tell you tumor necrosis factor there, which is a very familiar one that many people know of, and the sort of plaquing and all this that happens. This is a general review of the literature. T cells play a very important role as well. Keep your vitamin D levels up. So it's not accidental that the immune system will be playing a role, and improvements in T cells, which is good vitamin D and taurine, will actually get better results in the way the immune system will facilitate protection of the endothelium of the arteries. So that was just the sort of general cover. And anyway, that was just basically to outline what we're talking about. We're talking about the vascular smooth muscle cells, and they are very important in terms of, they are a very important element in terms of arterial calcification and arterial disease and stuff like that. So let's get on with it and look at the actual research that I want to show you. Taurine inhibits osteoblastic differentiation of vascular smooth muscle cells. So when we're talking osteoblastic, we're actually talking about calcification. Okay, so osteoblasts are basically also in the bones that actually input all the, those proteins that are actually produced and then activated by the menaquinones of K2. Anyway, so they are looking at how to inhibit the calcification and differentiation of vascular smooth muscle cells by the ERK pathway. Now, the ERK pathway is these are signaling type molecules at the outer part of the extracellular, the external part of the cell, and they communicate through these signaling molecules. They will communicate right through to the nucleus of the actual cell, play a very important role because they have to tell it what to do, so to transcript things, to start, constructing enzymes and stuff like that, and proteins to do certain things. So these are very important. So even in cell differentiation and stuff like that, trying to remember now. So basically, there's mitosis, when cell divides, they're involved in that. Then also when cells no longer can divide, they're involved in that. I can't remember what that's called. And there's also I think it's either called meiosis, and that is a certain type of cell division, but it's not. It's a different type. It's not your normal, where your cell divides and you create the two daughters that are related to the mother, the previous cell line. So, and they do all this sort of important signaling for a whole lot of these processes. They're also involved in terms of transferring phosphate within the actual, for ATP. So they do play a number of roles around in that regard. This sort of pathway, in particular, the kinase. So when you hear, usually they, when they talk in the general term, that it plays all these different sort of roles, they call it as protein kinase. In this case, we're calling it extracellular signal regulated protein kinase, ERK, because this is the specific type at the surface of the actual cell that actually signals right through to the nucleus. And this is where, remember the previous one that I showed you, the German Chinese study, where it actually was showing these, the cap and the sort of involvement of all these materials to build and calcify that and stabilize it and all that. Well, this is basically involved in all that signaling in that regard. That's sort of, and it's really important to basically be activated, because it keeps it downregulates some of the others that can be problematic. There's some of the other pathways. So let's get into it. So the abstract: vascular calcification develops within atherosclerotic lesions and results from a process similar to osteogenesis, so bone genesis. So you get lesions, you get damage, which we've talked about before, and then, LP little a moves in and sort of tries to stitch it up, calcifies it and stuff like that, to sort of stabilize that and all that. I've talked about it before. Taurine is a free B amino acid and plays an important physiological role in mammals. We have recently demonstrated that vascular smooth muscle cells express a functional taurine transporter. Got it? So this is really important, because up until now, there wasn't enough research. This was back in 2007. There wasn't a lot of research on you know, because there are taurine transporters all over the body in all tissues, but you know, there hadn't been any research to show that there was actually a taurine transporter in these vascular smooth muscle cells within the vascular tree, so within the arteries, capillaries, the veins, you know, the sort of the vascular tree, that there are these you know, taurine transporters. So taurine's playing a very important role, and so a deficiency in it means that certain things upregulate that have a calcifying effect. And when it's there, it's cleaning up the mess because it does have an antioxidant effect and all that, and it's down regulating that. So to evaluate the possible role of taurine in vascular calcification, we assess its effects on osteoblastic differentiation of vascular smooth muscle cells in vitro. The results show that taurine inhibited B glycosanophosphate induced osteoblastic differentiation of VSMCs, as evident by both the decreasing alkaline phosphate APL activity and expression of the core bond factor. Taurine also activated the extracellular signal regulated protein kinase, we'll just call it ERK to keep it easy, pathway. Inhibition of ERK pathway reversed the effects of taurine on ALP and CBFA1 expression. These results suggest that taurine inhibited osteoblastic differentiation of vascular cells via the ERK pathway. So this pathway keeps these ones under control, not to be overexpressed and causing calcification. And taurine is what actually supports this pathway. Not enough taurine inhibits this and allows this other stuff to happen and to increase vascular calcification. Okay, so, and I actually have seen another, I did see another study a while back. I just couldn't find, I don't know whether it's on my computer at work or where it is, but it actually did show sort of an association with high calcification and low taurine levels, which I found interesting, but it's an association. It wasn't causal. Here they've actually identified taurine transporter, so the actual enzyme inside these cells, and actually operating and interacting with key parts of the way cells divide, cells function, and stuff like that. So this is really important stuff. This is sort of, you know, sort of information. Vascular calcification, such as coronary and aortic calcification, that means that the aorta, you know, your valves and around there sort of can actually happen as well. So that can be a big problem. So it is associated with clinical complications such as myocardial infarction, impaired vascular tone, angioplastic dissection, and poor surgical outcomes. Exactly, poor surgical outcomes. And as you guys know, I've been discharged from my cardiologist. He just wants me to do a final test because pretty much, as he said to me, when you basically, you know, most people don't actually, because he knows that I sort of run up from the ground floor to the 9th floor. I do all these sort of things. That's just not normal. Most people that basically come out of heart surgery, bypass surgery in particular, like I had a double bypass, don't really recover well. They decline. That means they have damage to their heart, and that fibrosis of the heart just makes the heart work less efficiently, function less. It actually struggles and actually deteriorates over time. Well, the opposite has happened to me. That's what a species-appropriate diet, in particular taurine, is playing this really important role. And it's, you know, really got rid of all my fibrotic tissue. And that was my 2018 scan that I did. So that's now 4 years ago. And even in my lungs, it's brought down fibrotic tissue. It's really good at that. So, and that's another important thing, because a lot of fibrotic tissue is actually created in the actual vasculature where this damage actually happens as well. So it helps clear a lot of that up and reverse these sort of conditions. Previously, vascular calcification was considered to be a passive. So that's how they thought it was, just a passive, something that just happens as you get older. Yeah, I suppose with genetics, but it doesn't happen to our ancestors. Or then you got the bullshit because of the actual average life expectancy because of high infant mortality. No, I mean, they didn't live as long. Bullshit. You know, we only have to see Mary Ruddock next to it, a 122-year-old old woman who's jumping up and down. I can't even see a 50-year-old, and most 50-year-olds can't manage that, let alone have all her marbles as well, the old lady. So, I mean, give us a break. Absolutely nonsense. And they thought that it's degenerative end-stage process of vascular disease. It's just, you know, slowly just get worse. Everybody gets worse and all that. Yeah, if you're on a kibble diet, yes. Recently, however, the involvement of cellular activities in this condition has been recognized. Evidence suggests that several features of this condition are similar to those of skeletal tissue mineralization. Got it? Because of this calcification processes has similar features. It doesn't surprise me, because the body's going to use very similar mechanisms. They're going to use very similar enzymes and all that to do certain things. Why reinvent the wheel? You know, if you've got a bit of debt, you know, cover it up in this way. So these include the expression of alkaline phosphatase, ALP core binding factor A1, and osteocalcin. I've talked about osteocalcin in terms of MK4, you know, the sort of proteins that are involved in bone mineralization, and the osteopontin, and the presence of bone mineral hydroxyapatite and matrix vesicles. Vascular smooth muscle cells, pericytes, and macrophage in the vascular wall might be involved in the process of calcification. So there's a lot of things we still, you know, we can see something, but we've never had like an internal camera to see the whole process play out over a number of years. Obviously, you'd have to lock people up and monitor them in a very invasive way. Pericytes, and yep. Many experimental demonstrate that if vascular smooth muscle cells acquire the phenotype of osteoblast-like cells and played an important role in vascular calcification. Atherogenic inflammation mediated regulate osteoblastic differentiation of these cells. Therefore, these mediators might couple atherosclerosis and calcification. This strongly suggests that vascular calcification can be an actively regulated process. And that's what we're really learning, that it is. Taurine is a sulfur-containing free amino acid in mammals. Various physiological roles have been suggested for taurine, including calcium modulation. It regulates calcium, basically. I've talked about this before. Because it's an osmolyte. Membrane stabilization, a very important thing to stabilize membranes of tissue. That's where it actually controls the pressure. That was one of the most ancient things that taurine does, cell pressure. Intracellular osmosis regulation, which regulates the different, the fluids within the actual, which also has this effect in stabilizing the cell and managing the pressure role. Regulation of protein phosphorylation. Yes. We won't get into the phosphates and all that sort of stuff. And yes, you guys, I've talked about this before. Taurine is necessary for the normal development, and its deficiency leads to defects in growth, tissue differentiation, and immune development. So all these, this is what I talk about. Vitamin D and taurine for the immune system, cell differentiation, the fat-soluble vitamins, and taurine, growth, very important. We know that it's a myostatin inhibitor. We know that basically it's involved in a number of the growth pathways, and does work with leucine in that regard as well. So yes, a worldwide epidemiological study revealed that a strong inverse association between the levels of taurine excretion in the population, the mortality due to ischemic heart disease. And that's the one that I'd actually seen ages ago that actually showed these, that, not having enough taurine in your system seems to basically make things worse on that front. So it's the early stuff that I was actually reading and sort of looking. There wasn't, it was, but it made me drift towards taurine supplementation in those early days, which is, even though they were generally associational studies, all the research that I found in the subsequent times, mechanistic studies and others that show quite clear interactions with certain types of genes and other enzymes, and as a cofactor and stuff like that, and how it interacts with a number of systems. I'm glad I did get influenced by those, even though in hindsight, I would have been a bit less, I would have brushed them off in a sense, because they are, after all, epidemiological studies. They are looking at a single factor, so that does, but still, you can't credit, you can't identify cause and effect. So we've got to be very careful about that. Anyway, this suggests that taurine intake could be effective in preventing cardiovascular disease. And that was back in 2001. This other study in 2000 showed that taurine prevented the progression of atherosclerosis in rabbits. Remember the rabbit studies that really showed all the, problems with lipoproteins and all that, because they're just not adapted to certain fats and certain things that, they're very herbivorous. But even there, taurine was quite protective, indicating the probable therapeutic role of in atherosclerosis. Moreover, our previous study demonstrated taurine transporter, very important. The taurine-specific transporter was expressed on VSMC. Further evidence suggesting that taurine played an important role in the function of vascular smooth muscle cells by inhibiting their proliferation and preventing B glycerophosphate-induced calcification in these cells. Very important. However, it is still not known how taurine prevents the calcification of these cells. The objective of the present study was to determine the mechanism underlying this decreased calcification of calcifying VSMC mediated by taurine. Anyway, so they looked at, over a certain amount of time, did their experiments. And those were the results where they saw it has been recently shown that vascular calcification in vivo inside has many features in common with bone mineralization. Therefore, investigated whether bone associated molecules were altered during taurine treatment for mineralization. Alkaline phosphate is a well-established phenotype marker of osteoblast differentiation and a critical enzyme in calcification. They saw that being reduced. We'll go and look at that figure. So it showed that the dose and time-dependent effects of taurine on ALP activity in cultured VSMC cells after 12 days of culture and ALP activity in calcifying these groups dramatically increased compared to the non-calcified group, while taurine alone had no influence on the ALP activity, was induced by 5 millimoles. Taurine was lower than that of the calcified control. So they had a calcified control and one that had taurine in it. So the ALP marked decrease, while pretreatment of cells with ERK inhibitor blocked and decreased the ALP activity induced by the taurine. So let's go down, and you can actually see here, there's your taurine. And as you can see, the ALP is actually lower. Here's the calcium deposition. Again, that's without anything in there. This is with the sort of the inducing that. And you've got here the inducing, but with the taurine, it actually drops. Not to basically that, but it still reduces. Obviously, you know, minoquinone 4 is also something that will actually decalcify. So combination with fat-soluble vitamins, you'll probably get a more magnified effect. But still, it's playing a role. This is independent of fat-soluble vitamins completely. So really important. And so here, this expression, you can see that's quite statistically significant, the reduction. And here, basically, you have 0 taurine. Here you've got the thing that actually increases the sort of effect. So we're looking at the calcifying effect. And so the effects on taurine or CBFA1 expression in those cells in the calcification medium. So as you can see with the taurine, it does reduce it. Here it's got 0. Here, without that medium in there, look at the reduction with taurine alone. So if you're on a species appropriate diet and you're not basically doing a lot of derangements to yourself, you can basically really reduce things even further. This is why carnivores which are getting more taurine will have a dramatic change. Here is a person, theoretically model, that is basically, you know, doing something inappropriate that actually is increasing the calcification. And what actually happens is the taurine is reducing it. But when you take away the thing that's driving it, it dramatically changes, which tells you if you're on a species appropriate diet and you add taurine in there, you're going to get a much more magnified effect, a far better result. Here, they are not, you know, this is basically, this will show you that there is a reduction as well. Now, that looks different to that. That's with, you know, somebody, let's say, on a kibble diet, getting that effect, getting off the kibble diet with taurine, getting a dramatic reduction. Here's somebody who's left a kibble diet, but has actually got a calcification, and they put more taurine in, and over time, that will reduce the level of calcification. So that's how you interpret that. And that shows images of those sort of changes in the level of calcification, stuff like that. And that's pretty much it. I didn't want to go make it too long a video, but the key take-home message is that taurine will inhibit calcification in the vascular tree. So, which is really important that we reduce the amount and we can inhibit it. Now, obviously, species appropriate diet is very important. Adding taurine, adding minoquinone 4 as well, will help with that decalcifying process. And that's important. In the initial stages, my sort of view is to first allow the body to calcify, not to use basically high doses. You know, if you're in the early stages, you've just come across from a kibble diet, you've got certain calcification. If it's low, allow it to run its course. The body needs to sort of calcify to a certain level. Then when it's stabilized that tissue, you're no longer inflaming those arteries, then you can use this strategy to reduce. That means you go to high doses and then you start reducing and removing, and the body will clear. It's no longer inflamed. Your C-reactive protein or the other ones, inflammatory biomarkers are down. And that tells you, now I'm in the process of basically, let's clean it out. We're going to use taurine. We're going to use menaquinone 4 to clear it out. Also, you have to remember that you need to keep an eye on your calcium levels, because as calcium levels basically, like soft tissue calcification, drops and declines over time, what will happen is you need to bring the dose down of menaquinone 4, because, or raise the amount of calcium intake. Because what you can do is you can lower your calcium level, your serum calcium level. And the way you know that is that you're taking too much menaquinone 4 is when your heart's pounding out here. I actually had to comment on, I had an old comment that Chris Master John done on one of his videos, where somebody had actually asked him, of course, this was about 2 years ago, about menaquinone, about this sort of effect. And he said, oh, yeah, this guy was asking, is it menaquinone 4? And I just sort of answered, no, it's actually basically, you're losing, you're not getting enough calcium, because, it's probably, that person probably shouldn't be on those therapeutic doses of menaquinone 4, because they don't have calcium in their soft tissue. And if you don't have calcium, you're taking high doses, then that can be a problem. If you're not eating enough protein to bring in calcium, and you're not getting enough taurine to regulate calcium, then you could potentially cause yourself a subclinical deficiency in calcium. There's always that possibility. You need, because things work synergistically. I've said this over and over again. People need to stop thinking in a reductionist way and understand that all nutrients are working synergistically. And the doses that I put people on for menaquinone 4, for taurine, and many other things, some of them are outside the normal physiological ranges, and they're specifically for therapeutic reasons. They're not for general people. Let me make that quite clear. I constantly say this, but sometimes it seems like I'm talking in the wind and it's not getting traction. You've got to remember that if you're not sure, get to me, tell me your history, tell me everything, and I will tell you exactly the doses and how much, the period of time. And like the boron ones and all these other things, you need to follow my instructions correctly. Otherwise, you sometimes, otherwise go and do your own research. Don't bother me. I will tell you exactly, and the length of time you will take B vitamins or whatever, and you need to follow protocols specifically, so you don't put yourself. I don't tell you, oh, go and actually have Pfizer as much as you like. No, I know that it inhibits mTOR. There are a number of things that need to be taken into consideration when you actually give dosages or recommend anything in that regard. So, we want to improve things, but not make things. So if you're taking high doses of menaquinone 4, you need to ask your question, do I have a calcium issue? I can tell you, if you don't have soft tissue calcification, you'll know it, because your heart will pound like mad, because you're probably inducing yourself a reduction in serum calcium. So knock it off. There are lower doses, and you should be getting enough if you eat. And you should basically be, if you don't need it, you should be basically just getting it from the diet and eating things like brie and, camembert and things like that. There's plenty in that, in eggs and stuff like that. You do not need to supplement, or you can go for the low dose supplement that I also recommend, which is about 100 micrograms. You can take 2 of those, about 200 micrograms, which covers you perfectly in terms of your menaquinone 4 requirements. The 5 milligrams and 15 milligrams are specific for people who have specific conditions and may need these higher doses. For a short period of time, or for a long period of time, I'll set that parameter based on what their history is, what their condition is, and all that. Just keep that in mind. It's the same thing with taurine. My normal recommendations are usually about 2 grams, and anything above that, it's usually for therapeutic purposes of specific amounts. I think it's pretty safe, even up to 6 grams, personally. But I don't like people actually taking higher than that. And especially if they're on medications and other things like that, you really need to get back to me. Even if, look, sometimes I can take my time to answer emails, but you need to basically at least leave a comment and let me know, so you don't get yourself into any sort of problems and stuff like that. I've had a few issues with boron and a few other things with a number of people sort of taking higher doses and for a very lengthier period of time, and that can be a problem in that regard. Low calcium can also induce insomnia, which is another issue in itself. So keep that in mind if you've basically been playing around with too much minoquinone 4 and you're not getting enough calcium in your diet. That could be a problem. Some people getting a pounding heart, or some people get basically issues with the insomnia. So it can be a number of things. So, another one is pantothenic acid. Too much accumulation of that can actually cause insomnia issues as well. So there's a number of things that can cause issues. Too much blue light, a few things. So keep those things in mind, guys. This isn't sort of like, let me go crazy on taurine, like 10, 20 grams and all that. No, you need to basically, I always say to people, I want you first to calcify, to stabilize soft plaque, and then decalcify once your inflammatory markers have gone to. So when you're coming from a vegunarized diet or a SAD diet or whatever I want you not to be doing any of these strategies. I want you to basically only be doing about a couple of grams, like 2 grams or 2 to, just a couple of grams. If I want you to decalcify, I'm going to put you on 10 or 14, but I don't want you there. I want you to be on these lower doses for whether it's the athletic purpose or stuff like that, like I did with the other video, or for other purposes. That's fine for a short-term period to use certain types of strategies, but I don't want you on these high doses. I definitely don't want you on high doses minoquinone 4. I usually want people to stabilize their soft tissue calcification. And I usually recommend in those periods also that people consume dairy, specifically just to be protective of serum levels under those circumstances. That's the real thing. Now, we don't really have a lot of research. It's mostly based on a lot of anecdotes, unfortunately. But I have to always put the caution out there to remind people, because sometimes people forget and they like to experiment without talking to me before they do that. And so sometimes they get themselves into trouble. So don't be a naughty boy, behave yourselves, and you'll have less issues and you'll get the biohacks you want. So I hope you like this video. It's a different angle in terms of how taurine transports are regulating calcium within the vascular tree, and also injuries within the vascular tree. So it's a new way of viewing arterial calcification and stuff like that. A bit of a different twist that. And it sort of probably explains some extreme levels of calcification where you hear people having a CAC score of 1,000 or 1,500 or, these massive numbers. And it usually is when I've gone back and looked and talked to these people, I found that some of they were eating a lot of refined foods, which pretty much have got zero taurine. So they would have been for a decade or two, maybe even longer, on pretty much next to nearly no taurine or very little, whatever they were getting endogenously produced. And even then, they weren't eating enough methionine-rich foods, like, because methionine, cysteine to taurine, those enzymatic pathways. So you're not getting enough cysteine, you're not getting enough methionine. You're not going to be, producing a lot either in terms of taurine. So there's a lot of these sort of factors. So these people would have had severe calcification. When it's that high, I'm actually more willing to basically look at reversal strategies a bit earlier. But when it's much lower, like in the 400s and 300s and stuff like that, the thing that I have always concerned in my head is soft tissue calcification, where you've got basically still soft plaque, and that soft plaque has got a bit of calcification in it. It's not stable. That is the scariest stuff in my book. I'd rather have people very close to zero or very high. At least I know that's stable. That middle thing where it's sort of really low, 200, 300, 400, stuff like that, that's more of a concern to me than anything else. You know, I've even got people to use other strategies, sort of non-conventional strategies, to calcify more rapidly in order to stabilize that sign, you know, and then let's bring the inflammation down and then we'll decalcify you. So these things have to be done in their proper order. I've got another video that I did discussing that. I'll put it up here so people can go and take a look at that, because that's an important one to get your mindset in the right order as well. Anyway, hope you enjoyed it. See yous.